Hello, my name is Khadija, and I would just like to start off by saying that the extensive comparison of population DNA sequences is known as population genomics. Associated with population genetics is the neologism of population genomics. In order to better comprehend microevaluation and gain knowledge of a population's phylogenetic history and demography, population genomics investigates genome-wide impacts. While researching, I found that in a 2007 research, Megan et al. Compared the assembly of D. cuba and D. melanogaster to the complete genome sequence of many lines of Drosophila simulans. This was accomplished by first doing a whole genome study of polymorphisms and divergence before aligning DNA from D. Simulants whole genome shotgun sequences to a common reference sequence. This demonstrated that many proteins have undergone directional selection. They identified previously unidentified significant changes in divergence and polymorphisms along chromosomal arms. They discovered that X chromosome diverged more quickly and had substantially less polymorphisms than they had anticipated. Additionally, they also discovered areas of genome, such UTRS, that indicated adaptive evolution. I personally chose my project to be about population genomic analysis of sea urchin, Echinometra, SPEZ across an extreme environmental gradient, and how calculus plays a role in finding those population genomic analysis. While researching, I found that the most common genus of sea urchins, Echinometra, has been the subject of several research in ecology, speciation, and reproduction. However, the genetic information for this genus is often restricted to a very small number of chosen loci. For Echinometra SPEZ from the Persian or Arabian Gulf, it has been provided that a chromosome-level genome assembly based on a 10x genomic PAS-BIO and high-C sequencing with an N50 of 39.5 megabytes. The genome is assembled into 210 scaffolds, totaling 817.8 megabytes. The research estimated that ESPEZ genome to have 2N, which equals 42 chromosomes based on this assembly. According to the Busco study, which I found during my research, was that 95.3 of Busco genes were completed. 29,405 genes, including a conserved Hox cluster, were found by gene modeling and annotation using AB initio and transcript-informed data. ESPE, SPEZ gene families and transcription factors linked to environmental stress response, which is also known as defensum, were compared with other acneoide speciation, which with comparable high quality genome resource, since EZ may be found in high temperature and high salinity habitats. All species had roughly the same amount of defensome genes, but in ESPEZ non-coding regions next to genes implicated in pathways of environmental responsibility. It was also found that substantial indications of positive selection as well as losses of transcription factors, crucial for the environmental selection, as well as losses of transcription factors, crucial for environmental response. These findings offer important new understandings of biology of ESPEZ and the wider diversity of Echinometra, and will be an important resource for the community as they research issues with the taxonomic group and beyond. Extreme environmental gradients, such as great research platform for identifying the factors that influence patterns of genetic variation among populations. Furthermore, they enable more precise projections of the potential effects of upcoming environmental change on marine species. In contrast to the Gulf of Oman, which lies nearby by the Persian and Arabian Gulf, inhabits severe salinity, the temperature levels. Although the sea urchin Echinometra SPEZ lives in both of these seas and is a vital grazer and bioeroder for the coral reef health, no population genomic studies on this or other urchin species in this particular location have been conducted so far. The enormous population numbers, larger reproductive clutches, and extended lifespans of ESPEZ should, in principle, homogenize populations unless non-neutral activities are taking place, obviously. But I found that the researchers created a draft genome and a restriction site associated DNA sequences data set from the 7 populations in the Persian and, again, Arabian Gulf and the Gulf of Oman, separated by an environmental gradient. ESP-EZ's estimated genome size was 609 megabytes, and its 4.5% heterozygosity was among the highest ever found in echinoderm. From 85 individuals, it was extracted 918 high-quality SNPs that was used in further analysis throughout the research. Although there was population divergence and strong pairwise FST values along the two Cs, population structure analysis showed a very high degree of mixing between all locations, which also kind of concluded the whole research of the sea urchins. But numerous mathematical models are needed in order to comprehend and analyze the enormous amount of data generated by population genomics investigations. And as far as calculus goes, there are, as I just said, that there are numerous amount of ways where you can use to comprehend and analyze these data that are coming from population genomics, and we're always evolving, so it's just coming to even better ways to investigate that. And an area of DNA known as a qualitative trait locus, QTL, is linked to a specific phenotypic trait that fluctuates in severity and can be explained by polygenic effects, which are the results of 2 or more genes and their environment. These QTLs are frequently located on various chromosomes. The genetic makeup of a character is revealed by the number of QTLs that occur for a variation in the phenotypic trait. It might mean that a few large effects genes outnumber numerous small effect genes in determining, for example, like a plant height. In contrast to discrete characteristic, QTLs often underpin continuous traits. For example, traits that change over time, like height, for example. Traits that have 2 or several character values, for example, red hair in humans, a recessive trait, or smooth versus wrinkled peas used by Mendel in his experiments. QTL mapping is one approach or to evaluating this enormous amount of data, finding the genes causing adaptive traits that have been aided through QTL mapping. The fixation index, also known as the FST, is a metric that is used to measure the genetic variation within a population. FST has been used to demonstrate how selection affects a population when coupled with Tajima's D, because it is less vulnerable to changes in a species demography that would invalidate other selection tests. The McDonald's treatment test, also known as the MK test, which is very famous and is also used when looking for selection, with significant ramifications for many areas of plant biology research. High throughput sequencing technology has made it possible to analyze the many evolutionary factors that have produce the patterns of genetic variations that are present today. For example, the sea urchin that was based on this research. However, biologists now require expertise in information and statistics to manage such enormous amounts of sequencing data, because they don't want to mess up or have an invalid result. And the aim to explain population genomics methodologies to novices using a learning by doing methodology to assist the reader in independently analyzing the sequencing data is very important. In-depth descriptions of the evolutionary history of a particular species, for example, sea urchin for my project, or the discovery of genes selected by natural or artificial selection, are only 2 examples of the key evolutionary biology topics covered by conducted studies. And to kind of sum off, I feel like the extensive comparison of population DNA sequences, which was known as the population genomics, really kind of shows how the population genomic analysis of sea urchin echinometra space across an extreme environmental gradient reacts. And the conclusion that was based on that was that there was population divergence and strong pairwise FST values across both Cs. Population structure analysis showed a very high degree of mixing between all locations, and it wouldn't have been found without using the QTL methods or other like methods such as FSTs or McDonald's treatment like the MK test. Made it really easy to analyze those data So we could have conducted the experiment, which was to determine the population genomic analysis of sea urchins, Echinometra, SPEZ across an extreme environmental